There we go.
2 points per hit.
2 points per hit. Okay. All right, so round 2, we are going to now tackle chapter 4. Now, the reason why we're doing chapter 4 is because chapter 4 goes over cellular injury. So remember how I explained to you that disease process happens at a cellular level and then you work our way up. So what I'm going to do is go over injury to the cell, what happens, different types of injuries at the cell. And then we'll work our way into the next chapter, which is chapter 7, talking about neoplasias. So I think this is a really good kind of intro or interlude between getting into cancer cells and talking about cancer. Okay, and then we'll continue on with cancer next week. Okay, we'll talk about leukemia. Okay, so with cell injury, we do have 2 different types. We have what are called reversible cell injury and reversible cell injury. So we're going to start with the reversible type first. So when we talk about reversible cell injury, this means that the cell gets injured, it gets damaged, okay, whatever happens, but then whatever is going on, whatever process is going on with that cell, it can be reversed, okay, whether it's removing something or going back to its normal state. So the first type of reversible cell injury that can happen is something called hydropic swelling. Okay, so hydropic, I'm quite sure you may have heard of this term before, maybe not. But with hydropic swelling, this is when we have, say for example, a cell that actually starts to swell. Okay. Now what causes swelling inside of a cell or water that retains inside the cell is when we have faulty sodium-potassium pumps that happen on the channels of the membrane. Okay. And as you know, if we have a faulty sodium-potassium pump and if these ions just kind of go haywire, and let's just say, for example, we have an influx of sodium, we know what is the next thing that it's going to attract is what? Water. Okay, very good, water. So as we have more sodium, now water is going to be attracted, and this water will just get absorbed in the cell, making the cell swell. Okay, so this is one of the first signs of cellular swelling. Now they say here that what makes these sodium-potassium pumps go haywire is the fact that ATP is not there, okay, because these pumps are driven by ATP. So ATP not being sufficient enough to get these pumps or open up these channels. You open the door. To open these channels to make sure that these ions do get inside the cell, okay. Now, what do you know on a cellular level that makes ATP? Thank you, glucose. Somebody said that. Okay, glucose. Okay, keep in mind that glucose, and we're not talking about blood glucose, but glucose on a cellular level is used because, as you know, it goes through the whole citric acid cycle, electron transport, okay, all that stuff that we don't want to remember. No. But keep in mind that those processes are extremely important because the end result is that we have this energy in the form of ATP, and it is so important that we have it in order to have our cell membranes functioning as it should. Okay, so if for some reason there's not enough ATP, not enough energy, those channels or pumps go haywire and now causing an influx of sodium, now causing an influx of water, hence swelling. Now, this is a picture of, or a good example of hydropic swelling. Now, the first side of this slide over here, this histology slide, is showing us, and let me just kind of do a review first. This is normal. So, just to kind of refresh your memory from anatomy, okay, what type of epithelial cell is this? What type of cell?
Simple squamous.
Not so much squamous.
No.
Cuboidal. Okay, cuboidal. I always tell my students, look at the nucleus, because usually cuboidal has a very nice round, okay, nice plump looking nucleus. Okay, so make sure that you always look at that. So this is cuboidal. This is what we call simple cuboidal. Now, these cells here, simple cuboidal cells, what type of tissue is this, if you remember from anatomy? If you've had anatomy here, I'm quite sure Dr. Youssef showed you this slide. No? Where do you find most simple cuboidal cells? Do you remember? This is the kidney. Okay, this is the kidney. So, simple cuboidal cells, did you say that? Okay, very good. Kidney. Okay, so most simple cuboidal cells are definitely found in the kidney. Okay. Now, this is normal, and then what they're showing you over here, if you look at this particular simple cuboidal cell, or this kidney tubule, I should say, you can see that now these cells are actually blown out. Okay, they look large, and not only do they look large, but they're not as nice and uniform as this side. You can see that it almost looks like there's some sort of disruption even over here with the membrane, which, let me just state that these cells we're talking about here for reversible swelling, but not so much a disruption of membrane. Obviously, you know, if the membrane is disrupted, that's irreversible. So in any event, they're showing you here the swelling, and not only that, if you look at the lumen here of this kidney tubule, it's actually decreased, okay, because of so much swelling that has occurred. So that's a very good example of showing you hydropic swelling. Okay, so with hydropic swelling, not only just water just takes up all of that space in the cytoplasm and just make it really large, but what also happens is that the organelles also get large as well. So the water also gets an influx of water inside the organelles, the mitochondria. Okay, so everything that's inside endoplasmic reticulum, all of that will get large. Keep in mind that when we talk about cellular swelling, okay, just like we're taking the whole level of organization, the tissue swells, the organ swells, okay? And when we get this organ swelling or getting larger, the clinical term for that is what is called a megaly. Anytime you hear the term megaly, this is a situation that is what we call an abnormal size, or usually an increased size of an organ, that most of the times come from the fact of some sort of imbalance or disease or condition. Now, very good example is cardiomegaly, okay? So with cardiomegaly, just to give you an example, even though it doesn't, well, kind of talks about hydropic swelling a little bit. But say, for example, you have a patient that has not enough blood that's flowing through the body. Okay, so decrease perfusion of blood. So low perfusion of blood, and so what's going to happen is that if the person does not have enough blood flowing through to the different organs and different parts of the body, what's going to happen is the body's compensatory mechanism is to get the heart to pump faster. Because the more the heart pumps, the more blood is going to get pumped out and faster to the different areas of the body. So what happens with that is as the heart pumps faster and faster and faster, what's going to happen is the cell damage that's going to occur to those muscular cells are going to be this hydropic swelling. It will get larger. Now the muscle tissue gets larger, and now we have a cardiomegaly. The heart gets larger. And the heart will now go outside of its boundaries. And most of the times we know this is definitely from the contraction of the heart, because the heart is a muscle. And as it continues to contract, it'll just get larger and larger. So just keep in mind that, and especially with a cardiomegaly like that situation, that is abnormal. It is an abnormal process that happens, and it's due to some sort of condition that the body is going through. Now, another thing here is that in the cells, sometimes we can have what are called intracellular accumulations. Okay, so what does that mean? So we all know that the cell is like a little organ in itself. Okay, so it has all the different organelles that do have different functions. And we know, say for example, lysosomes. Okay, so we know that lysosomes are there for what activity? What is the function of a lysosome? Okay, so break down any type of debris, or let's say anything that should be digested, okay, and to get rid of. Now say for example, if lysosomes do not function, if lysosomes aren't functioning properly, now we have just a bunch of material that's just sitting inside the cell that is not broken down. So this is what we call intracellular accumulations. So now we have an excess, and what happens here is that now the cell is not going to be able to function as it should. Now we have a condition, if you so to speak, with that particular cell, and then which can lead to further things. Now, the other thing too with intracellular accumulation, sometimes they can be accumulate from secondary to faulty metabolism or a synthesis. Okay, very good example that I'm going to go over with you, and they have here common site liver. Very good example is the liver. Liver has many functions. Okay, so what is the main function of the liver that you know? One main function of the liver.
Besides alcohol? No.
Okay. Well, that is the main function. Is detoxification. Let's just say that. The liver is our main detoxification organ. Okay, so yes, if you go out the night of drinking and enjoying yourself, okay, what happens is that the large amount of alcohol that you ingest in your body, what your body is going to do is take that alcohol, which is a toxic substance, and convert it into a non-toxic substance. Okay, So that is what the liver does. And thank God the liver does that, you know, because that's the only organ that will take care of that. Now, what are some other functions of the liver that you know? Liver has some other functions. Releases bile, very good, which means that it is also part of the whole fat metabolism, okay, process. Because of the bile, and the bile we know helps break down in part of the fat lipid metabolism process. Very good. So let's stop there. What happens now is, say for example, alcoholics. Okay, so people that are alcoholics, what happens here with alcoholics, the body is now, with the liver, is now focusing so much on the alcohol and going into a detoxification process continuously that now the lipid metabolism goes secondary. So what will happen is that these individuals will now start to accumulate lipids in their liver, okay? And I'm quite sure, or maybe you heard of it, maybe not, people that are alcoholics sometimes develop what is called fatty liver, okay? And this is because the detoxification is now taking precedence over all of the other functions that the liver should be doing. So that's one very good example of intracellular accumulation. Now, they're showing you a little picture here, and I apologize you can't see this in the back, but they're showing you just a few more of these cellular injuries and what can happen. Okay, so abnormal metabolism can occur, and again, we use the example of a person as an alcoholic. So metabolism goes haywire because of the fact that alcoholism is taking over. Now, another thing here is protein mutation. Sometimes, as you know, proteins are extremely important and needed for DNA, okay, process. And so what happens here sometimes, if for some reason there is a mutation that occurs with the DNA, this will obviously now cause what we have an abnormal cell, okay? This abnormal cell can now lead to other issues, okay? Very good example of this would be, as you know, some genetic disorders, okay, that children can have just because of the fact of different mutations. The other thing here is enzymes. As you all know with our body, and we talked about lysosomes being the enzymes that are needed to break down certain particles and things inside the cell. But keep in mind that if there are no enzymes, okay, present, or if your body is lacking an enzyme, it will not be able to break down that particular situation. So, very good example that I use all the time is people that are lactose intolerant, people that are truly lactose intolerant, okay? These individuals do not have the enzyme lactase. So what happens with lactase, they don't have lactase, so what will happen is these individuals will just have an overproduction or an accumulation, so to speak, of lactose, just sitting in, and I'm just using an example as a cell. But in the GI tract, lactose will just sit there in the GI wall, okay? And what happens here is that the body does try to break it down even though the enzyme is not present. So the way of breaking it down, it actually turns it into a gaseous state, okay, because of the type of compound it is. It'll turn into a gaseous state. Hence the reason why if anyone's lactose intolerant, you don't want to be around them when they take their little, if they haven't taken their little pill. Because they do have an accumulation of gas, okay, and it only comes from the fact that their body's just trying to break down that lactose, okay, so it's not their fault. Okay? So that's that. Now, the other thing here is indigestion of indigestible materials. Okay, so what is that? That is, say for example, and I'll show you a very good picture in a little bit. Say for example, a person that works maybe in a factory, okay, that are maybe doing something where they're soldering and different metals and things, that unfortunately they may inhale, okay, even though they're maybe wearing protective gear, but somehow they inhale this particular particle and it gets trapped in the lungs, okay? Now what happens here is that when they inhale this type of particle, the body does not have the enzyme to break down, say, coal or silica, but what will happen here is that it'll just get stuck and it'll just stay there. And as it stays there, obviously it will cause damage. But keep in mind the immune system will try to do its best to get rid of it. So that's just a good example of that. Okay, this is just another slide showing you the fatty liver. Okay, you don't have to worry about this too much, but just in case if you're interested, I don't know if you took a look at during histology a normal liver, but the difference between a normal liver and a fatty liver are these vacuoles here. So you see these large white circles. These are vacuoles. These vacuoles are normally present in the liver, but they're actually way smaller than that. Okay, so this is a person with fatty liver, so what happens here, the fat just accumulates and just gets all deposited in that area of the vacuoles. Oh, what did I just do? Oh. This picture here, I don't like. You can cross that out. Helping us out anymore. Okay. Now, this is a picture, I talked about this already, someone that has, this is a person that has silicone lung, okay? So this person inhaled the silicone. Now keep in mind that we will go over an entire chapter on respiratory diseases and definitely go over occupational diseases. And so again, what you're looking at here is you see all of this blue area, kind of like a bluish blackish area. This person here now has inhaled, okay, silicone, and it is now just these dust particles are actually just deposited in their lung fields, okay. Now, just to kind of pick your brain and just to see where you're at as far as physiology and the process. If this person has this silicone dust particles in their lung, what do you think the body is going to do at this point? What system do you think is going to try to come in and try to attack it? Okay, the immune system. The immune system is going to come in and try to attack it because obviously we do not have an enzyme to break it down, hence the reason why it got trapped in the lungs in the first place. Now, the other thing here is once the immune system comes in and try to attack it, so say for example, we know macrophages, they're going to come and try to take it away. Now, if we have a large infiltration of macrophages getting to this area of the lung field, what other things do you think is going to happen from that area? Or what would be, I would say, an outcome from macrophages just trying to go in and get rid of it? Not so much of a fluid accumulation. They could have fluid accumulation, maybe just from some of the chemical mediators and production of that part of inflammatory fluid. But what is the macrophages going to do? They're going to try to eat the silicone dust, but while they're eating the silicone dust, what else are they eating? They're eating the lung itself, and they're going to start eating the epithelium. So what's going to happen is those macrophages are going to come and just kind of, they're doing what they're supposed to do because they're trying to get rid of the abnormality. But the problem here is that these macrophages are going to come in, they're going to try to take the epithelium or eat away the epithelium, and now the epithelium is going to be very thin, and they'll break it. And then also what's going to happen is now a result of that could also be scar tissue and other type of infiltrates and things that can just be stuck in the lung, okay? So keep in mind, and this is why I was explaining to you earlier, that the immune system plays a large part in a lot of the disease processes. I would say if you're not that strong with the immune system from physiology or your previous courses, just make sure you kind of just reread that chapter and just kind of fresh up on it if you need to. Okay. Now, another thing we're going to talk about is cellular adaptation. Okay, so sometimes these cells can go into different changes, okay? So whether we're talking about size, shape, or if we're talking about number of cells, okay? So the number of cells that you can produce. So the first type of cellular adaptation that we're going to talk about is atrophy. So atrophy, I'm quite sure you've heard of this term before. Atrophy is going to be a decrease in the cell size, so a shrinkage, okay, that can occur. And this can come from a number of factors, okay. Disuse is one very good example, is muscle. So obviously if you have a patient that is bedridden and they're just sitting in the hospital bed for weeks and months at a time, obviously their muscles will go into a state of atrophy because they're not using them. Denervation and ischemia. Keep in mind that nerve and vascular supply is extremely important for any organ in the body, okay? So if these individuals are not getting that type of supply, and even nutrition, and when I mean nutrition, starvation, we're talking about, if we're using muscle, for example, we know that what makes muscle contraction happen? What ions make muscle contraction happen?
Calcium.
Calcium. What else? Potassium. Okay, so keep in mind that any ion that is important with muscle contraction, this is what we're talking about here with nutrient starvation. Okay, so blood, nerve, nutrients. and interruption of endocrine signals and persistent cell injury. So there's a lot of different things that can cause any type of cell, muscle, tissue, what have you, go into a state of atrophy. And also aging. As we do get older in the aging process, this is a normal situation that can happen, okay? Unfortunately, yes, things do start to get a little smaller, okay, and things do start to go south, okay, because we start to lose loose connective tissue and collagen and all that good stuff. So, yes, something to look forward to. So, atrophy, yes, does happen normal. Now, normal cell, they're showing you a picture here. Then we have atrophy, which is a decrease in cell size. Another thing here we'll talk about is hypertrophy, increase in cell size. Hyperplasia is an increase in cell number, not size, but just the increase in cell number, and that's due to the increased mitosis. Then we have what is called metaplasia. Metaplasia is a conversion of one cell type to another. Okay, so one cell type to another. And just to let you know, if something like this goes on, I don't want to say it's a red flag, but it's something that you should monitor. And then definitely if we have a dysplasia, where all of a sudden you have a very nice normal looking cell, and then you have these cells that are just all clumped together and you don't know what it is, what's going on, is it squamous, is it cuboidal, you don't know what shape it is. This definitely tells you that this patient at a stage of a dysplasia is considered pre-neoplastic. So this is precancerous. This is something that is extremely important and it should be monitored. Okay, not even monitored, but they would even go further by doing biopsies and all that other stuff. And making sure that this is taken care of, and not only taken care of, whether they cut it out, freeze it out, or whatever they do as far as treatment, but also making sure that this does not come back. Okay, so this is very important.
What did you call it?
Pre-neoplastic. Pre-neoplastic also meaning precancerous. Okay, so let's take each one of these individually. So I showed you hypertrophy. So with hypertrophy, this is an increase in cell size. Okay, so we already know what hypertrophy is. So now there is two different types of hypertrophy. Okay, one I already went over with you. So say, for example, we have physiological hypertrophy and then pathophysiological hypertrophy. Physiological hypertrophy is, say, for example, you go to what is it, LA Fitness, and you're getting it in, okay, working it out with all the weights, and you come out looking like that, okay? Now, that is physiological hypertrophy. So your muscles are definitely, when you see these big buff guys, okay, yes, their muscles are in a state of hypertrophy. Now, what happens is that that's physiological hypertrophy, and that's normal physiological process for what he's doing, because he worked very hard to get his body, okay, to that point. Now, when we talk about pathophysiological hypertrophy, this is when the example I gave you as the heart contracting, okay, and contracting very fastly to try to compensate for the lack of blood flow going through the body. This is a pathophysiological hypertrophy, and that's when we get the cardiomegaly, and the heart gets very large. So that's not a normal process. Okay, so there's a difference.
So one is bad and one is good?
Yeah, so physiological hypertrophy that's a normal process. So like I gave you the example with the guy in the gym. And then pathophysiological is the abnormal. Okay, that's abnormal. So, because we don't want to say all hypertrophy is bad. Okay, so some hypertrophy is normal. Okay, depending on the situation. Okay, so they're showing you here a picture of cardiomegaly, or if you want to say hypertrophy. And here they're showing you the difference between hypertrophy, and this, as you can tell, definitely looks like the left side of the heart. So here this person has hypertrophy in the left chamber, here on the left side of the heart, okay? And they're showing you here about 9 centimeters, whereas this normal heart is now at 5 centimeters. So you can see how large it is also by looking at how small the lumen is. So you see that it's a very thick muscle mass there. Okay. Another way to also detect, and you guys will go over this, I'm quite sure later on, is making sure you know the boundaries of the heart, and I believe you went over that maybe in anatomy, knowing that the heart sits between ribs 2 through 7. And then what will happen is sometimes even through imaging you can see this cardiomegaly. or sometimes even through auscultation, okay, that you can feel, okay, and outside, or know that the heart is outside its boundaries. Okay. Hyperplasia. With hyperplasia, this is going to be an increase of number of cells, excuse me, an increase in the number of cells, not the size. So mitotic division gets activated at this point, or elevated, okay, when we're talking about a hyperplasia. So you probably wonder, okay, well, how do you get a hyperplasia? What causes this? There are a number of things. They do say sometimes hormones can do this. So as we know with hormonal situations, say for example, maybe thyroid disease or things of that nature, where people do have an imbalance, can cause certain organs and things to get thicker or larger. Another good example that I have a picture of on e-college is callus formation. Okay, that's a very good example of hyperplasia. So people wonder, how do you get calluses? So what happens with that is the epithelial cells, okay, are constantly being irritated from maybe your shoes or your rubbing or whatever it is that you're doing with your feet. And so this constant irritation to the epithelium will now cause the mitosis to get activated and increase. And so now you have an increase in number of cells in that area because it's constantly being injured. So what happens here is that now you have a thickening just in that area. Okay, so this is what happens with hyperplasia. Metaplasia. I have a very good example of metaplasia for you smokers in the room. Okay, don't raise your hand. Now, respiratory, or let's say the respiratory epithelium is what type of epithelium that you remember from anatomy? Respiratory epithelium is what type of epithelium in your throat? Stratified, squamous, ciliated. Okay. Now keep in mind that this is normal. With smokers, and over time as they smoke, what happens is that that cigarette smoke burns off the cilia. So that ciliated epithelium now turns to a non-ciliated. Okay, so that is a cellular adaptation. The good news is if a person stops smoking, yes, okay, it can regenerate and turn back.
How long does it take?
Huh?
How long does it take?
It could take probably a couple of months, weeks to months. What you'll notice is when people do smoke, because they don't have cilia, sometimes they always have kind of like this chronic cough, especially smokers, that seasoned smokers, so to speak. They do have a chronic cough because, you know, the cilia is there to sweep out any type of debris or whatever, but they don't have it. The other thing too is that once your body, once you stop smoking and your body is now regenerating back to its normal epithelium, once the cilia comes, it will actually now start to remove all of that nasty debris, okay, that's there. So then people do have more of a cough actually when they stop smoking. But it lasts just for a little bit. It doesn't stay that way forever until their respiratory tract really gets back to where it needs to be. Okay, so that is one good example of a metaplasia. Okay, and that's common. It happens. I remember my girlfriend, when she stopped smoking, she coughed and coughed. And she was actually coughing up a lot of phlegm, too, because all of that gook is being released, finally. Okay. Now, dysplasia. Dysplasia, again, this is when we have a normal cell, and then all of a sudden it just goes haywire, okay? Now, again, when you look at, and we'll talk about this again later next week when we go into leukemias, when you see cells that are to a point where they're disorganized, you can't differentiate what they are, and especially if they're in a cluster format, that is definitely not a good sign. We don't want to say, oh, that's cancer, but it's definitely, if it's not cancer, it is sort of pre-neoplastic or possibly leading to that direction. So this is neoplasia. Okay, and let me just show you really quickly the pictures that I have. I know I'm out of the camera view, but it's okay. So I just want to show you muscular hypertrophy. I do have here and atrophy. Hyperplasia with the foot, okay. Metaplasia, and let me just show you really quickly what I'm trying to point out here with metaplasia. With metaplasia, here is a simple squamous cell, and then all of a sudden these cells are now turning into a cuboidal. This is what they're trying to show you here. And then here with a dysplasia, this is squamous cell, and then also now showing you the squamous cell going into a state of a dysplasia where this is just now, we don't even know what it is, okay? Usually when you see something like this that's pre-neoplastic or neoplastic, you'll see the nuclei very large, and you won't be able to see much of the surrounding or the rest of the organelles or the structure of the cell. So that's another determinant factor that is maybe along the lines of cancer too. This is actually cervical dysplasia, just to let you know what you're looking at. Cervical dysplasia, I used to work in a GYN office in undergrad, and actually kind of had a lot of that cervical dysplasia in the office, and so I remember the procedure they would do to get rid of that. I don't know if they still do this. They would do a cryo procedure to try to freeze out the cells in that area to try to get rid of it, or they would do this procedure called a loop with some sort of instrument to just kind of pull out the epithelium of that region, and then obviously go back and recheck and make sure that these cells don't come back. So they do try to go in and take care of that situation. I'm going to fall on camera. I'm recording. Okay.
Should we put some bushes there too?
I know. I know. And that's the problem. My mom, she tells me, she's like, you're such a klutz. She's like, I don't know how you do anything. You're such a klutz. And I am. I don't know what to do. Okay. So now we're going to talk about irreversible cell injury. Okay. Now let me go back to, sorry. Let me just go back to this really quickly and let me just explain something. Atrophy, hyperplasia, hypertrophy, and even metaplasia, all of those cellular adaptations are reversible. When we get to a dysplasia, dysplasia is irreversible. So I want you to understand that dysplasia is irreversible. I don't want you to think that it is reversible. It's irreversible because at this stage, if it's that disorderly, it's not going to go back, okay, to its normal size or shape. The only way to get rid of that, as like I told you, is any kind of procedure, okay, to remove the cells. Okay, now with irreversible, just continuing on, we do have to talk about necrosis, okay? So you guys may know, just common knowledge of necrosis or something necrotic. What happens when something gets necrotic? It dies, okay? So this is when we're talking about not only cell death, but tissue death, okay? And if it's really bad, organ, okay, the whole organ death, if it gets that far. So keep in mind that when something's necrotic, this is usually from the fact that there is not enough blood supply getting to that particular area, okay, or the tissue. So when we have, say for example, the cell not getting its proper blood supply, okay, or nutrition and what have you, what's going to happen is that the cell will start to die. And one of the ways that the cell will start to die is that the organelles that are inside the cell will almost start to sort of disintegrate, okay? And what will happen is it will start to just kind of fizzle out and ooze over, okay? Hence the reason why if you ever see something necrotic, it's very hard, okay, and looks like it's just kind of withered away. Or sometimes if you see necrosis, it can be wet and it looks almost like somebody poured acid on it, okay, and it just looks like it fizzled.